Welcome, everyone, to our very first lecture on insect vector and diseases. Today, we are diving into the foundational topic of insect classification and identification. You might think that telling one tiny bug from another is just a matter of academic curiosity, but in the realm of medical zoology, it is absolutely critical. By the end of this session, you will understand exactly why we classify insects and how we use precise mathematical tools to identify them correctly. To begin our journey, I want you to imagine walking into a massive, chaotic library where millions of books are just dumped on the floor in no particular order. If you needed to find one specific book on treating a rare disease, it would be nearly impossible to locate it quickly. This is exactly what the natural world looks like, with over 1 million different types of insects buzzing and crawling around us. To make sense of this overwhelming chaos, scientists created a highly organized filing system, much like sorting books by genre, author, and title. This system allows us to bring order to the natural world. Now that we understand the need for a system, let us look at how it actually works. In biology, this sorting system is called classification. It is the formal scientific process of categorizing insects into a hierarchical system based on their evolutionary relationships and shared physical traits. We start broad with the class Insecta and narrow down through orders, families, genera, and finally, species. Identification, on the other hand, is the practical application of this system. It is the process of figuring out exactly which book you are holding. To do this, entomologists use a tool called a dichotomous key, which guides the user step by step using paired physical descriptions to find the correct species. Building on the idea of dichotomous keys, let us explore what we actually look at when identifying an insect. We cannot just glance at a bug and guess. We must examine specific anatomical traits. Key identification features often include the type of mouth parts, which tell us how the insect feeds. We also look closely at the structure of the antennae. Furthermore, the number and type of wings, along with the specific arrangement of veins on those wings, act like a fingerprint for the insect. Understanding these physical features allows us to accurately place an insect into its correct taxonomic group, ensuring we know exactly what we are dealing with. You might be wondering why we need to be so incredibly precise about naming a tiny bug. Well, in the study of insect vectors and diseases, accurate identification is literally a matter of life and death for human populations. For example, only specific species of Anopheles mosquitoes can transmit malaria, while Aedes mosquitoes transmit dengue fever. If a public health worker misidentifies the mosquito species in a village, they might apply the wrong type of insecticide or implement a completely ineffective control strategy. This could lead to a devastating disease outbreak. Knowing the exact classification tells us about the insect's biology and weaknesses, allowing us to target it efficiently. Understanding the high stakes involved, we realize that we cannot rely on mere visual estimates. In taxonomy, we do not just look at insects and guess, we measure them precisely to distinguish between closely related species. This brings us to a common mathematical tool used in insect identification known as a morphological index. A morphological index takes a visual shape, like how round or long a head is, and turns it into a hard, objective number. By relying on mathematics, we remove human error and subjectivity from the identification process, ensuring our public health data is perfectly accurate. Now that we know why we need objective numbers, Let us look at our first mathematical tool, the cephalic index. Conceptually, this formula gives us a standardized ratio that describes the shape of the head, regardless of the overall size of the insect. The formula is CI equals W divided by L, all multiplied by 100. Here, CI represents the cephalic index, which is a dimensionless number expressed as a percentage. W represents the maximum width of the insect's head in millimeters, and L represents the maximum length of the head in millimeters. If the width increases while the length stays the same, the index increases, indicating a broader head. Conversely, if the length increases, the index decreases, indicating a longer, narrower head. Let us see this formula in action with a concrete example. A medical entomologist captures two different mosquito larvae. For larva A, we are given a head width of 1.2 millimeters and a head length of 1.5 millimeters. We are asked to calculate the cephalic index. We chose this formula to objectively describe the head shape. First, we substitute the width and length into our equation, placing 1.2 in the numerator and 1.5 in the denominator. Dividing these gives us 0.8. Multiplying by 100 gives us a final cephalic index of 80. Physically, a value of 80 clearly indicates that the head is longer than it is wide, giving us a standardized metric to compare against taxonomic databases. Following our analysis of the first specimen, let us calculate the index for the second potential vector, larva B. We are given that larva B has a head width of 1.4 millimeters and a head length of 1.4 millimeters. Again, we substitute these values into our formula. Notice that the measurements are identical for both dimensions. Dividing 1.4 by 1.4 yields exactly 1.0. Multiplying by 100 gives us a cephalic index of 100. Physically, an index of exactly 100 means the head is perfectly symmetrical in terms of its width and length, indicating a perfectly round or square head. This mathematically clean result is highly significant for our identification process. Now that we have calculated the indices for both larvae, let us interpret what these numbers actually mean. We found that larva A has a cephalic index of 80, meaning it has a relatively narrow head. In contrast, larva B has an index of 100, indicating a perfectly round or square head. In practical taxonomy, this significant numerical difference confirms that these are two distinct species. This distinction is absolutely crucial if one species is a known disease vector and the other is completely harmless. By relying on math rather than just visual guessing, we ensure our public health data is perfectly accurate and our interventions are targeted correctly. Having seen how head shape helps identify mosquitoes, let us look at another critical disease vector, the sandfly. Sandflies are notorious vectors of a severe disease called leishmaniasis. Because they are incredibly tiny, identifying them requires looking at microscopic details, specifically the ratio of specific veins on their wings. Just like the cephalic index, calculating a wing ratio transforms a visual observation under a microscope into a hard, objective number. This allows taxonomists to confidently determine the specific subgenus of the sandfly, which is essential for tracking and controlling the spread of leishmaniasis. To identify these sandflies, we use a specific mathematical relationship called the wing ratio. Conceptually, this formula compares the length of a specific branch of a wing vein to the stalk it branches from. The formula is WR equals R2 divided by R2 plus 3. Here, WR stands for the wing ratio, which is our final taxonomic metric. R2 represents the length of the second radial vein on the insect's wing, measured in millimeters. R2 plus 3 represents the length of the combined radial vein stalk before it branches, also in millimeters. If the R2 vein is significantly longer than the stalk, the ratio increases, which is a key indicator for certain dangerous subgenera. Let us apply this formula to a specimen. Suppose a taxonomist measures vein R2 as 0.8 millimeters and vein R2 plus 3 as 0.4 millimeters. We are asked to find the wing ratio to determine the subgenus. We substitute the given measurements into our equation, placing 0.8 in the numerator and 0.4 in the denominator. It is vital that both are in millimeters, so the units cancel out. Dividing 0.8 by 0.4 gives us a final wing ratio of 2.0. Physically, a ratio of 2.0 or higher often points to the subgenus Phlebotomus, confirming this specific insect is highly capable of transmitting leishmaniasis to humans. To fully grasp how we get these numbers, let us look at this diagram showing the exact morphological measurement points. On the left, you see the schematic of an insect's head. The horizontal dashed line, labeled W, shows exactly where we measure the maximum width, while the vertical dashed line, labeled L, shows the maximum length. These are the exact dimensions we used for the cephalic index. On the right, we have an insect wing. Notice the specific veins highlighted by the blue arrows. The upper arrow shows the R2 vein length, and the lower arrow shows the R2 plus 3 stalk length before it branches. This diagram teaches us exactly where to place our microscopic measuring tools to gather the data needed for our formulas. We have covered a lot of ground today, so let us review our key takeaways. First, we learned that insect classification and identification form the foundational bedrock of medical entomology. Without it, we cannot distinguish dangerous disease vectors from harmless bugs. Second, we saw how dichotomous keys use specific physical traits to guide us to the correct species. Finally, we discovered that mathematical indices, like the cephalic index and wing ratio, transform visual observations into hard, objective data. Mastering these techniques is your first critical step in understanding how to combat vector-borne diseases. You now have the foundational knowledge to approach insect identification scientifically. Great work today!